Hello everyone, my name is Majid and today's topic is about male reproductive toxicity. I will be discussing the meaning of reproductive toxicity or reprotoxic effects, as well as the aims, strategies and the guidelines for designing reproductive toxicity studies. In 1956, a drug named thalidomide was introduced into market in Germany and some European countries as a safe sedative. And since the studies showed that it was impossible to achieve lethal dose of thalidomide, they were able to market this drug as an OTC drug. And therefore it became very popular with pregnant women for the treatment of morning sickness. Now at that time, no one knew that thalidomide had a serious mutagenic effect. And that was the start of later to be called the thalidomide disaster or tragedy. This tragedy affected more than 10,000 families, around 2,000 babies died, and around 8,000 had to live with serious disabilities. Only 5 years later, in 1961, it was proposed that thalidomide could be the cause of those incidents. And this disaster shocked the world, and everyone realized that more regulations are needed to prevent such incidents from happening in the future. And many scientists believe that if there had been more extensive testings on laboratory animals before the drug was marketed, this disaster could have been avoided. In 1968, the WHO Program of International Drug Monitoring was formed, and since then, more regulatory authorities and organizations have been established throughout the world, all to prevent such disasters from happening again. In general terms, reproductive toxicity refers to the structural and functional alterations that can affect reproductive competence in sexually mature males and females, and also the adverse effect on their offsprings and their development. Some documentations make a distinction between reproductive toxicity and developmental toxicity as two separate categories of toxicity. However, for the purpose of this seminar, we will include both categories under reproductive toxicity. We can see here some of the classes of reproductive toxicities, such as male and female fertility, which include toxicities such as damage to the reproductive organs or alteration in the endocrine regulation of gametes and their maturation and release, as well as reduction in sperm count in cases of male, and overall reduction in fertility for both males and females. And it's also included under this class the alteration of mating behavior and the ability to mate. Another class of toxicity, which has to do with parturition and childbirth, it includes the toxicities that affect labor and delivery, which include abnormal or difficult delivery, and the changes in the onset and or duration of the birth. Some drugs also can affect lactation, and they can alter the process of lactation in the nursing mother. For example, the quantity or the quality of the milk can be altered, or the overall maternal behavior toward nursing the offsprings. Other classes which mainly has to do with the development of the offsprings include classes such as mortality. In this class, the positive signal can appear as abortion or stillbirth or loss of implantation, neonatal death. And some drugs can also cause dysmorphogenesis, which means the effects that are seen as malformations or variations in the skeleton or soft tissues of the offspring, commonly referred to as structural abnormalities. Drugs can also affect the growth of the offspring or cause a functional impairment. For each class, there is a different assessment. For example, the body weight is the most common measurement for assessing growth rate. And in case of functional impairment, the common assessments include locomotor activity and the time to sexual maturation and the mating behavior and fertility of the offspring. Next is the ICH, which is the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. So it brought together the regulatory authorities of Europe, Japan, and US, and it included also experts in pharmaceutical industry from these three regions. And the main objective of the ICH was to develop a process targeted to or aim to reduce or prevent the need to duplicate testings conducted during the research and development of new drugs. And this was by recommending new ways to achieve more harmonization and the requirements for the product registration. So when there is a standard guidelines in doing certain experiments or testings, it's most likely that the results will be all consistent. So this will mean no need to duplicate already done studies, and that is the general idea. And this will lead to more economical use of human, animal, and material resources, since less study has to be done, less duplicate studies. This means less animals and humans and materials will be needed for that particular need. It also led to the elimination of unnecessary delay in the global development of new drugs, and of course the availability of quality and safety and efficacy of new drugs, since they are all are developed according to the proposed guidelines. And that also led to the enforcing of more regulatory obligations in order to protect the public health. Now we come to the general idea behind designing and performing reproductive toxicity tests. It's perceived and thought that animals which are treated during defined stages of reproduction will better reflect human exposure to that product, and it will also allow more specific identification of what stages are at risk from using that product. But while this approach might be useful for most medicines, some medicines will only cause toxic effects over long-term exposure to low doses. So these are better represented by 1 or 2 generation study approach. So the actual testing strategy should be determined by the anticipated drug use, the desired use of the drug and for what it will be used, as well as the dosage form and route of administration intended for humans. But we also have to make use of already existing data on that product or medicine being tested, such as toxicity effects and pharmacokinetics and pharmacodynamics, as well as the similarities to other compounds in structure or in activity. This means if drug A being tested is similar to drug B, and drug B is already known to cause certain toxic effects, when analyzing drug A, we will be looking or anticipating the same toxic effects that is produced by drug B. Now this doesn't always have to be the case, but it is a useful approach when analyzing or testing new drugs. Now, to apply these points successfully, flexibility is needed. As mentioned by the FDA guidelines, there is no guideline that can provide sufficient information to cover all possible cases. So that means that all persons involved in the test or study should be willing to discuss and consider variations in test strategy in respect with the ethical standards in human and animal experimentations. So the aim of reproduction toxicity studies is to reveal any effect of one or more active substances to mammalian reproduction. For this purpose, the investigation and interpretation of the result should be in relation with all other pharmacological and toxicological data available to determine whether potential reproductive risk or toxicity in humans is greater or lesser or equal than the other toxicological manifestations. Also, repeated dose toxicity studies can provide important information regarding the potential effect on reproduction and particularly on man fertility. So the prime goal of repeated dose toxicity study is to characterize the adverse toxicological effect which will occur in result of repeated daily dosing or exposure to that medicine for a specified period of time. So in support of initiating human clinical trials, this study is required. First, this study should be done before a clinical trial study can be approved. And the length of this type of study, it depends on the expected length of treatment in humans. And when data is acquired from a study, we have to assess the relevance to humans, to extrapolate the results to humans. And to do that, data on comparative kinetics and mechanisms of reproductive toxicity might be useful for that. And whatever combination of studies selected, it should allow exposure of mature adults at all stages of development, from conception to sexual maturity, and to allow detection of immediate and latent effects of exposure. Observations should be continued through one complete life cycle. That means from the conception in one generation through conception in the following generation. Following are the ICH stages of development. We will be discussing each stage in details in coming slides. They are defined from A to F as follows. A is the pre-mating to conception stage. B, conception to implantation stage. C, implantation to closure of the hard palate stage. D, closure of the hard palate to the end of pregnancy. E, birth to weaning. And F, weaning to sexual maturity. Now, this classification into stages is important because certain reprotoxic effects can be observed only during certain stages of development. One obvious example of that, let's say a drug has a sexual dysfunction reprotoxic effect in men, or it causes loss of libido. This can only be observed during the pre-mating to conception stage. That's the only stage we can observe if that's really happening. Stage A, pre-mating to conception. This stage evaluates reproductive functions in adult males and females. These functions include development and maturation of gametes, mating behavior, and fertilization. Now it's obvious that we should know how pregnancy would be timed. Different species will have different gestation days, and we should know when to start counting the days. So by convention, it was agreed that pregnancy would be timed on the basis of either when a spermatozoa are identified in a vaginal smear or a copulatory plug or mating plug is observed, and that they will identify the gestation day GD0. And that is the case even if mating happened overnight. Now, the aberrations that we can detect during this stage include the effect on libido, the loss of libido, which is a common effect of alcohol intake. And we can also observe impaired sperm motility during this stage, which is a common effect of caffeine. We can also observe decreased sperm count and female reproduction cycle disruptions. Some examples of that are the anti-malarial drug Cosibol, which causes a fertilization disruption, and some steroid hormones. They disrupt egg release. Ovulatory blockade, ovarian dysfunction, and also ZIDO pregnancy can also be observed during this stage. Stage B, conception to implantation. Now this stage examines reproductive function in the adult female and pre-implantation and implantation stages of the conceptus. That is what the embryo in the uterus is called in the early stages of pregnancy. In rats, mice, and rabbits, implantation occurs in 6 to 7 days of pregnancy. And the detectable aberrations here include male-mediated developmental toxicity. Now, animal studies in the 80s and the 90s suggested that genetic damage after radiation or chemical exposure might be transmitted to offsprings, but it only came to public attention or awareness in the 1990s, when it was reported that the men that were working at Sellafield, which was a nuclear site, might be causing leukemia in their children. Another abnormality that we can detect during this stage is failure of desudilization, which is a process that results in certain changes in the cells of the endometrium, which is in preparation for or during pregnancy, including morphological and functional changes. Now, some studies suggest that failure of desudilization will result in facilitating the implantation, but higher chances of pregnancy failure. During this stage, we can also detect failure of implantation and ectopic pregnancy, which is a complication of pregnancy when the embryo is attached outside the uterus, which can be fatal if not treated in time. For example, the fallopian tube can burst, causing internal bleeding. In fact, 3 to 4% of pregnancy-related deaths are due to ectopic pregnancy. Now, there is some drugs that are known to affect implantations. These drugs include some antihistamines and a hormone named diethylstilbestrol, which is an non-steroidal form of estrogen. Now, this hormone usually used as a estrogen replacement for the treatment of estrogen deficiency, which normally means that it will aid in pregnancy. However, some studies suggest that women that were already exposed to this hormone will have less chances of successful pregnancy using in vitro fertilization.
Here we can see a schematic representation of the study used to evaluate fertility and early embryonic development to implantation according to the ICH guidelines. This is the study to evaluate stages A and B, and this is the study that we will be discussing in details later, as it's the main one that has to do with male reproductive toxicity.
Stage C, the stage between implantation to closure of the hard palate. So embryogenesis is completed when the closure of the hard palate occurs, which considered to be 15 to 18 days through pregnancy in rats, mice, and hamsters by convention, and 30 days in guinea pigs. So during this stage, we can evaluate some reproductive functions of the adult female, as well as the embryonic development and major organ formations. The detectable aberrations here include post-implantation loss, which is calculated by determining the ratio of dead to total implants from the treated group in compared to the ratio of dead to total implants from the vehicle or control group. This morphogenesis and the formation of abnormal tissues can also be observed during this stage, as well as growth retardation. We can also detect dominant lethality during this stage. A dominant lethal mutation is a mutation that occurs in germ cells. It results in the death of the fertilized egg or the developing embryo, which can lead also to spontaneous abortion when pregnancy is lost without any external influences, or when the embryo is delivered before it can survive independently. Mutagenesis and the generation of genetic mutations can also be observed or detected during this stage.
During stage D, we can still evaluate the adult female reproductive functions, as well as fetal development and organ development and growth of the offspring. The detectable aberrations here include spontaneous abortion and growth retardation and premature delivery, which can be also detected in the previous stage. But here we can also detect interference with histogenesis, which affect the differentiation into cells into specialized tissues and organs during the growth of the offspring. The limitation or impairment of physical abilities can also be detected here, as well as toxemia, which means sometimes there will be some toxins or infections or poisoning that can be detected by blood tests. Here we can see some examples of substances or drugs that can affect the fetal development or cause malformations. After birth, we can still evaluate the female reproductive functions of the mother, as well as the adaptation of the neonate to extrauterine life, such as pre-weaning development and the growth of the neonate. The detectable aberrations here include any difficulties that can occur during delivery, as well as any growth retardation to the offspring, any interference with lactation. As we have mentioned before, some medicines or drug or substances can affect the quantity or quality of the milk, as well as failure to thrive, which means that the newborn does not meet the recognized standards of growth. That means that newborn should be examined regularly to evaluate growth as well as the CNS functions. And if there is any morphological CNS disruptions, it can be also evaluated during this stage. Any abnormal maternal behavior that is seen should be recorded and evaluated as well. The last stage is between weaning and sexual maturity, stage F. Now this stage is generally treated only when the substance to be tested is intended for use in children. However, some guidelines recognize that it might be sometimes appropriate to treat the neonate during this stage, in addition to continuous evaluation of the offspring. This stage provides observations on post-weaning development and growth, adaptation, and the attainment of full sexual development. Whether the offsprings were treated or not, or it's just a latent effects caused by the exposure of the mother to the substance. Most of the aberrations that we can see here are related to the sexual growth or sexual maturity of the offspring. Such aberrations include cases like cryptorchidism, a condition in which both or one of the testes fail to descend from the abdomen into the scrotum, and hypospadias, a congenital condition in male in which the opening of the urethra is on the underside of the penis. Delayed maturation or early puberty can also be seen. And here we can see some examples of medicines and substances that can affect birth, lactation, and sexual maturation. Here is the schematic representations of the studies used to evaluate stages C to D and E to F. They mainly have to do with female reproductive toxicity and fetal development, so these parts will be covered by my friend in the female reproductive toxicity seminar. As mentioned before, in order to evaluate certain reprotoxic effects of a certain substance, we have to evaluate that substance during the appropriate stage, and we have three types of studies as suggested by the ICH guidelines to evaluate reprotoxic effects. Each study has to do with different stages. So in order to do a comprehensive reprotoxic study, we have to combine studies. And here we can see a schematic representation of a study that combines stages from A to D. So according to the study to be performed, a discussion has to be made on what studies to combine and how to do so. So as mentioned before, this test is to evaluate toxic effects resulting from treatment from before mating till implantation. So this should involve stages A and B for both males and females. And it should assist the maturation of gametes as well as mating behavior and fertility for both males and females. For females, it will also evaluate the pre-implantation stages of the embryo as well as the implantation. Now we only need to use one species of animal in this study, preferably rats, because of the accumulated knowledge we have on rats. And there is no specific number of how many animals we should include in each group in the guidelines. However, a generally accepted number is 10 males or 12 to 13 females per group. And doses are not also specified because each substance will have different dose to toxicity ratio. Therefore, the dose level might be based on information from acute toxicity tests or from repeated dose studies. Generally, at least 3 test groups should be used with different doses, and the highest dose used should be intended to cause the maximum toxicity and not to cause death. Now it's advised by the guidelines that the mating ratio should be 1:1, and some separation procedure should be there to allow the identification of both parents of a litter. As we can see in the diagram here, the treatment for males should start 28 days before mating and 14 days for females. It's mentioned in the guidelines that male animals can be sacrificed any day after conception is confirmed. However, a generally accepted waiting period is 14 days before doing an autopsy. The observations during the study include checking general signs and mortalities among the animals at least once daily. Body weight and body weight changes should be checked at least twice weekly. We should also monitor how much food animals are consuming, and this should be done at least once weekly. Now, if this is a combined study that includes females as well, a vaginal smear should be done daily, at least during the mating period, to determine whether there is an effect on mating or precoital time. The observations at terminal examination include necropsy and macroscopic examination of all adults. Organs should be preserved with macroscopic findings for possible histological evaluation, and sufficient organs from the control group should also be preserved for comparison. Testes, epididymis, ovaries, and uteri from all animals should be preserved for histological examination, and the tissue can be discarded after completion and reporting of the study. For male animals, we should also record the sperm count in the epididymis or testes, as well as sperm viability.